And those are mostly focused on the ability of carbon dioxide to be carried by the blood, and in fact, some of it being carried by hemoglobin itself, but the majority being carried as bicarbonate. And that brings us to the last topic of a negative feedback regulation of respiratory rate and depth. So, so these are all part of the last essay. And the last essay we've covered part of because we have discussed the Bohr and Haldane effect, but it's going to come back, the Bohr and Haldane effect, when we discuss the role of a critical enzyme, carbonic anhydrase, today. So I would like to get started. I know we still are a bit underrepresented by people, but that's their issue. Remember that on Thursday, you've got a pile of stuff due. So these lecture questions that we're finishing up now are due on Thursday by 5 p.m. And the spirometry handout is due by 5 p.m. So in lab today, we will discuss the spirometry handout, make sure that you understand what happened in those tests and why you do those tests. And then the rest of the time is for you to finish up your histology drawings if you haven't on the respiratory system. Make sure you know what you're labeling, what you're supposed to label, have it all finished so that you can turn it in today by 5 p.m. So that's just the histology part. And if you have questions on any of the models, then I'm here to answer those as well during the lab, because we will be doing the lab exam next Tuesday, a week from today. And what I'm planning for the lab on Thursday is to start the urinary system. So it will not be an open lab for you to study and ask questions. That's kind of today. That way we won't fall behind in the urinary system. Okay, so we're slowly getting more folks here, but I would like to recap. Well, let's go back to lecture question 18. And I don't think we actually got this group to cover this, but it has been talked about previously, so I thought I would cover it. And then we did have 3 groups set up for today. The first group will be talking just about how CO2 is transported and this key enzyme, carbonic anhydrase. The second group will be talking about bicarbonate, the Bohr effect, and indirectly Haldane may come up as well. And then the last groups, well, actually there's 4 groups, are talking about the negative feedback and regulation and what other factors affect respiratory rate and depth. So
Can I interrupt you real quickly, Professor? Apparently there was another link to another Zoom session, and I went into that first, and there's a couple people over in that other room.
Oh, you know, that was my mistake. It said 8 to 9:30 on it.
Yeah.
So should I go over there? You're thinking, I don't know if I can do 2 Zoom sessions.
Yeah, I was Sarah and somebody else was over there, so okay,
yeah, that was my setting up my sessions for office hours starting at 8 in the wrong shell. Okay, let me I'll leave this. Let me see if I can open another Zoom session. In the past, I couldn't, but maybe now I can. Let's see what happens if I go over there. Well, so it's ended. Oh, this stuff isn't on, that's why. Okay. Dashboard. Okay. Hey, you guys, I apologize. I accidentally put my office hour one in here, but we're over in another Zoom session. So
You're
if you
still
guys
with us.
go with you, so maybe I can see them. Maybe I can chat to them.
Yeah.
So I see them here, but maybe I can chat to them. Hopefully this will get to them. Did you see it, Dina? The chat?
Well, I closed out of it so I could join this one.
I know. Are you seeing a chat in the one you're in now?
No.
Oh, good. All right. So they just left. All right. I can now end this session, which shouldn't have existed. Okay. Zoomed again. What can I say? All right. So let me see. They'll probably take a second to get here. There was just 2 people left in that session. So mostly we've done announcements. There's Louis. Okay, sorry about that, Louis, and Sarah, if you're here too. I accidentally put my office hour one into the wrong shell. Okay, so what I'd like to do is recap a little bit on the lecture question 18, which is around BPG, and then we'll start with putting you guys into groups, and we're almost all here now. So I actually did a little research on BPG since I've seen conflicting things in our textbook at different times. It is true. Oh, thank you, Sarah. I'm so sorry you got confused where you're going, but that was my fault. So BPG is apparently, and this is what I've researched, I'm not going to test you on this per se. The question just asks, what is BPG? And it is a byproduct, this is something we've talked about, of anaerobic respiration. So glycolysis could go along its normal pathway of blood cells and just make the ATP, the 2 ATP you get out of it in the end, and acetyl-CoA. Or it could go on a side pathway where it makes less ATP, and one of the end products is BPG. When it does that, BPG can bind to hemoglobin and lower its affinity. Now, the conditions under which it does that will cause unloading of the oxygen, and that tends to happen more out in the tissues. BPG levels then drop in the lungs, and therefore indirectly help the loading of oxygen by not being bound to hemoglobin. That's what we've said before. That's what was talked about back when we talked about red blood cells and earlier. So that's really the answer to that question 18. So the little bit I read about it online is that there's actually again, I won't test you on this, but there's actually 2 isomeric forms of BPG. And what happens during low oxygen tension conditions, like in the tissues, is there's an enzyme that converts the one isomer into the other. And the one that it converts it into is the one that will easily bind hemoglobin. When it's out in higher oxygen, like lungs, it doesn't secrete as much of that enzyme. Therefore, you don't have as much BPG. But BPG is always being made at some level. It's whether you're converting it or not. So I think that's, again, I won't ask you about that. But the reason I thought that was interesting is it's one of the ways in which our body adjusts when you have chronic low oxygen tension, like people who move to high altitude. they tend to express more of the enzyme, and therefore they find it easier to unload more oxygen from their hemoglobin than somebody living at sea level. And the other reason I thought it was interesting was because fetal hemoglobin, which we haven't talked about much in our list here, but fetal hemoglobin won't bind the BPG, and therefore it tends to generally have it's one of the reasons it has higher affinity for oxygen than the mother's hemoglobin. Because after all, when the mother's hemoglobin is out in the placenta, it's basically under a tissue situation, and oxygen is being transferred to the baby's hemoglobin. So there's more than one reason the affinity of fetal hemoglobin is higher, and therefore oxygen jumps ship from mom's hemoglobin to the baby's hemoglobin. But part of that is the BPG and that's converted doesn't occur in the fetal hemoglobin. It's not sensitive to that, at least. It won't bind BPG. Therefore, it's more readily able to pick up oxygen. So I just thought that was kind of interesting, and does relate to something that's, I don't think, in our list of lecture questions, but that fetal hemoglobin has a higher affinity to oxygen than adult hemoglobin, and that was one of the reasons. So, are there any questions on BPG, your answer to BPG, before we break into our groups? Okay, it looks like almost everybody is here. There's always a few missing folks, but all right. If you do come up with something, and we'll hopefully have some review time in lecture. If not in lecture, then we can do some in lab today. So let me break you up into 4 groups. And again, I'll put the lecture questions on those groups. So 4 groups, like I said and I'll sign manually, create, and we'll rename this A, and actually questions 19 and 20. And we'll just assign some people here. Okay, and then we'll rename this one B. Questions 21 and 22. Okay, yes, and we'll assign. Okay, and we'll do this one. C, and it's question 23. And yes, and we'll assign maybe. I have to assign some of you guys back into the other rooms. Too many people in the other 2, 4, 5. Okay. Oops. D, and question 24, 25. All right. Yes, and assign. Maybe not. We have 5, 5, and 6, so to save one person. How did that happen? Stacy, and maybe we'll assign to this one, the last person. Okay, here we go. So I'm going to change it to being just, whoops, I hit the wrong thing. Just to being 10 minutes because you only have a couple questions. So if you can please make sure you have your spokesperson chosen. And if you want to break it up to 2 spokespeople, that's fine as well. I don't think anyone has more than 2 questions. So I'm going to go ahead and open the rooms for 10 minutes.
Hi guys.
Still not. Okay, folks, I think most people are back. So do we have a spokesperson for Group A?
Yeah, that's me, Professor Manry.
Okay, Manpreet, how about that question 19?
Okay, so for the modes of transportation for carbon dioxide, for 7 to 10% is dissolved in gas since CO2 is highly soluble. 20% is bound to hemoglobin, but the majority of carbon dioxide is converted into bicarb, which is 70%. So that's what we put for question 19. And then we described like the whole entire process of turning it into bicarb in question 20.
All right, I'm going to share a screen so we can see this enzymatic reaction that it, as you're talking about it. And then I actually have the picture of the red blood cell if we need to. If that made it easier for you, we can share that too. Oops, what did I do? Here it is. Hopefully you're seeing that screen. So here's the reaction that we're talking about. Ca stands for carbonic anhydrase, and it's fine if you use that abbreviation as well. You notice the book does. So go ahead, Manpreet, you can talk about this enzyme.
So the body starts with carbon dioxide and water, and it's converted into carbonic acid, which is quickly with the use of the enzyme carbonic anhydrase, which is Ca. Carbonic acid is weak and thus is ready to lose a hydrogen bond or gain some. So from carbonic acid, it will turn into bicarbonate and hydrogen. The bicarb will be released from the red blood cell in order to go into the plasma and travel to the lungs. So to exit the red blood cell, it will use a secondary active transporter, which allows...
We could probably hold off on that just because the next question that's their sort of answer. But we just talk about what happens, which way do these go in the lung versus in the tissues? If I were to make arrows, which direction of this balance would go towards in the lungs versus in the tissues?
Yeah, so for the tissues, I believe the carbon will turn into carbonate, right?
Carbonic acid. Mm-hmm.
Yeah, and then it'll turn into carbonate and hydrogen.
Yeah, so in the tissues, your waste product is CO2, right?
Mm-hmm.
And like any balanced equation, if you add onto this side, this is a balanced equation. If you're adding onto this side, it's going to push the equation this direction, right? This is in the tissues. And that's what she just said. It's going to cause more substrate to be around for this enzyme. And there's always water, so you don't have to worry about that. So you add more CO2 and the balance gets pushed in this direction, which means that H plus is increasing and bicarbonate is increasing. Now she started to talk about the fate of this bicarbonate, and we will come back to that in the next question. So if we were to contrast that with in the lungs, then what would happen, Manri?
So it would just go the opposite way. So the bicarb and hydrogen would turn back into carbonic acid and thus turn back into carbon dioxide and be expelled.
Exactly. Now, my recommendation to you is to know this very, very well, because we're not going to just finish it with this exam. And the next, we'll go off into the kidney for a while, but eventually we're going to come back to this because we're going to be focusing in on how the body regulates its pH. And you'll see briefly when we get down to the last couple lecture questions that you're going to use your respiratory system to help you not just get rid of CO2, get oxygen, but also help you regulate pH, because after all, this is pH, right? The concentration of H plus. And when we talk about regulating blood pH, we're also actually talking about regulating all the body fluids pH, since the blood is in communication with that. So eventually we're going to come back to this in great detail. Again, a little bit aside, because I'm saying stress this, really know this equation. I kind of made a summary, which I could probably share with you guys later, of all the things that carbonic anhydrase is going to be doing over the next remainder of the semester. So the black over here is what's happening in the red blood cell. And we'll come back in more detail to that shortly. And then, as I mentioned, it's useful, the carbonic anhydrase is useful in the regulation of blood pH by your respiratory system, which we'll touch on briefly in the last couple lecture questions. And then we're going to do the kidney. And in the kidney, you'll see that carbonic anhydrase is required for you to bring bicarbonate back into the blood from what's filtered out as you make your urine. And then if you have pH imbalance, the kidney also uses the enzyme to help you balance that pH, either too much H plus or too much bicarbonate. It's going to use that enzyme to help you do that. And then when we get in the digestive system, we're going to reuse this enzyme in order to secrete H plus and to retain bicarbonate. And that bicarbonate is going to eventually be used to help neutralize the stuff leaving your stomach and going into your intestines. So why I'm putting up this up here is not because you need to know all this on the test, but rather to strengthen your interest and hopefully your concentration on this enzyme, since it's going to be so important. And not only will it be important, but this equation will be important, this little bit of chemistry. Now notice that this enzyme works in both directions, but it's always doing the same thing. It's either making CO2 and water into carbonic acid, or it's taking carbonic acid and making it into CO2 and water, but it will catalyze both directions, and that's key to this. All right, with that bit of an aside, did you have anything else, Manri? I mean, I know you wanted to talk about what's happening to this, but that's the next question. So
I just wanted to add that this process also happens in the plasma, but it will be without the enzyme, so it'll be much slower, so there won't be as much carbonate made.
Brilliant. Okay, so this is kind of something you can relate to. CO2, when it dissolves in water, I think I brought this up last time, It does spontaneously without this enzyme, but very slowly form carbonic acid, dissociate and make H+, and that's one reason why carbonated beverages have that little bit of bite in in your taste, and maybe clear your palate a little bit, your saliva if you're thirsty, because it's low in pH, it's acidic. And so that will happen very slowly. Thank you. That was great to add. So that means that when you add CO2 and generate CO2 in your tissues, and you start pushing the equation this way, carbonic anhydrase is helping you convert CO2 that's being carried in your plasma as a dissolved gas. Initially, some of that dissolved gas is, in fact, the majority is converted into this stuff, into bicarbonate. Some remains, that 7% that she mentioned, and some of the CO2 that you generate ends up being bound to deoxyhemoglobin to make carbaminohemoglobin. All right, so how does it go from this dissolved gas through this equation and end up as bicarbonate? This would all be in the intercellular fluid. And the next question talks about why the levels in the plasma are rising. So who is the spokesperson for Group B?
It's going to be me, Stacy, for 21. I'm not sure if it's correct, but from what I understood, the bicarbonate levels in the plasma are rising in the tissues because the offloading of carbon dioxide is much quicker and it enters the red blood cells faster due to carbonic anhydrase. Mm-hmm. And what this enzyme does, it catalyzes the conversion of carbon dioxide and water to carbonic acid, and it decreases in the lungs because it's essentially reversed back into carbon and water in the lungs by having bicarbonate re-enter the red blood cells and bind with hydrogen.
Okay, well, you're perfectly right in what you said, but we still have the bicarbonate stuck in the intercellular fluid when you're out in the tissues. So how does it get from the intercellular fluid when you're in the tissues into the plasma? This is if you don't know, then Manri, you can go ahead and finish what you were going to say about the transporter.
Yeah, I can say that. So it basically has to do with the chloride shift, and that's when bicarb is exiting and chloride is entering. And it's chloride because it's the most common anion in the extracellular matrix, and it will electrically balance the cell as well. And that's using a secondary active transporter.
Right. So this transporter is secondary active not because of the sodium potassium pump. Here's one of the cases where you have a secondary active transporter that is not getting its gradient from the sodium potassium pump. It's getting its gradient because carbonic anhydrase makes so much, it uses ATP to do it. It makes so much bicarbonate. You get a high level of bicarbonate into this intercellular fluid when you're out in the tissues. Now, what you have on the other side in the extracellular fluid is high levels of chloride because, of course, chloride is your major anion in your extracellular fluids. It's got a single negative charge. Bicarbonate has a single negative charge. To keep charge balance, this antiporter switches so that the chloride moves in and the bicarbonate goes out. So that's called the chloride shift. So here, whoops, sorry, I'm almost hitting that because it's right at the bottom, right where I want to put my hand since I'm right-handed. So chloride in tissues, chloride exits, chloride enters the cell, sorry. So we'll say one chloride enters, that's supposed to be a negative, for each bicarbonate that exits. The red blood cell. Okay, now what happens in the when you go into the I did the colors wrong, but what happens when you go into the lungs? Remember, in the lungs, you're going to be pulling the bicarbonate and combining with H+, and this is all has to happen in the intercellular fluid. So in order to have enough bicarbonate to keep this going so you can keep expelling your CO2, what are you going to have to do with the chloride shift this time? Anybody?
It's just going to reverse.
It is, right. So this antiporter is a dual purpose, just like the carbonic anhydrase was. So the red is in the lungs. You're going to go the other way. So now for the lungs, you're going to reverse this. In lungs, one bicarbonate enters per chloride exiting. Now, some earlier editions, they called that the reverse chloride shift. I think this book just calls it the chloride shift again. But I don't care, whichever way makes sense to you. If it makes sense to remember it, that you are shifting chloride out versus shifting chloride in. So chloride comes into the cell and the tissues and then exits the cell in the lungs, and it's married to the bicarbonate shift. So then you can keep pulling more bicarbonate out of the blood, turning it into CO2 when you're in the lung and expelling it out into the environment. Are we okay with any questions on that? Okay, so what is the role of the Bohr effect in this one? That is question 22. Is that still you, Stacy?
No, I'm going to answer, Professor.
Okay, let me just share the screen showing that picture because, oops, let's pull it up. So that might help you when you're talking.
Okay, thank you. So if I relate this to the Bohr effect, to the role of the enzyme CA in the tissue due to the pressure gradient when carbon dioxide is picked up and oxygen is released with this enzyme, with the help of the enzyme, it causes the generation of hydrogen, and then hydrogen will bind to hemoglobin, which is the Bohr effect. Because in the Bohr effect, at least they should have one hydrogen bond and less than 4 oxygen.
Okay, great. So what she's showing is we're following this side of it. CO2 is coming in. It's causing there to be a lot of substrate for this carbonic anhydrase. That means you're pushing the equation in this direction. Bicarbonate's going up. Here it goes out with the chloride shift, but H plus also goes up. As H plus goes up, more of it will bind to hemoglobin. That will boot off an oxygen, and you will unload oxygen by the Bohr effect here. Now, I added haldane just to remind you, once you've booted off that oxygen, then the hemoglobin is able to bind CO2 to the globin part of it. So it can't bind CO2 unless it has already lost one or more oxygens. So then what happens, I started to go there, in the lung is kind of the opposite, right?
Right.
Is that the next one? No, I don't really ask you that one, but yeah, so here you have the equation going this way because CO2 is being pulled out by that gradient, and as it's pulled out, it tends to deplete the substrates for the carbonic anhydrase, pulling the H plus off of the hemoglobin, therefore raising the affinity so oxygen can bind and we become oxyhemoglobin again. This also does show what Manpreet brought up, that in the plasma, just in any water solution, you have this happen slowly. So the width of the arrows is meant to represent that most of the CO2 that is generated from bicarbonate out here that had to come in through that chloride shift and then be converted by carbonic anhydrase. Less is carried by the as a carboaminohemoglobin, and then the dissolved gas in the plasma is even less, 7 to 10% as a dissolved gas. Okay, great. So thank you, guys. Let's shift gears here, and I don't think I had anything else on that, but if there are any questions, this is a good time to ask it on this, because we are shifting towards regulation. Let me go to chat just to see if there's, because I don't see chat right now, see if anything's popped up. Okay, all right, so so anyway, there's something in chat, but I'll come back to that. It's not for everyone. All right, so let's go on to this new topic and maybe just showing you a little bit of the anatomy since I have this open. Are you seeing this new slide of the anatomy of the regulation of the respiratory rate and depth?
Yes.
Yeah, great. All right, so I want to remind you, and I know there was a little confusion about this in the last exam, that these carotid aortic bodies have two types of sensory cells. I believe they are totally different sensory cells. I don't know for sure that that's the case, but my guess is they are, because one is a baroreceptor. It detects pressure, and we talked about that in the regulation of blood pressure, and they adapt rapidly. The others are chemoreceptors, and they are the peripheral receptors looking at a variety of features in the plasma. These are not, let's just assume, they're not the same cells as the baroreceptors, because they actually are going to send their information to a different location. They're not going to send it to the cardiovascular center where the baroreceptors sent it. They're going to send their information up here into the medulla. So in the medulla, we're going to have two groups of nuclei, and it's fine if you want to call this one the VRG and the other one the DRG. And it's referring to whether their position ventral versus dorsal group. So the question asks, explain why PCO2, the partial pressure of CO2 in blood plasma, affects central receptors, but blood pH does not. Now, where these central receptors are distributed apparently through the brainstem. So there isn't a picture here showing you where central receptors are, because they apparently are distributed through the brainstem, so that you're monitoring your pH and indirectly your PCO2, or CO2 indirectly, the pH maybe is a better way of putting it, throughout your whole brain, and that's going to be sent to the ventral respiratory group, the VRG. So who is the spokesperson for group C that wants to talk about these central receptors?
Hi, that's going to be me, Professor Prabdi.
Okay.
Okay. So pretty much what we got was just a that the PCO2 in blood plasma can affect central receptors because CO2 can cross the blood-brain barrier into the ISF, whereas the H plus ions can't, meaning that the pH can't directly affect central receptors, but it can affect it indirectly. And then what we said for the peripheral, all I got was that because sorry, I have it written down. I lost my place. Okay, so all I really got was that because they can, the peripheral receptors can directly detect the PCO2, and they can also detect, or they can also independently detect the pH.
Great. All right, so let me stop sharing this for a moment and we'll go back to the screen because I want to help you with this essay since I think it's a good big part of the essay that is somewhat, it's all in the book, but it's somewhat distributed. It's not really put together very well in one location for you to kind of read about that. Central receptors, just like she said, are sitting in cerebral spinal fluid. So these are in the CSF, right? And CSF is unbuffered. And this is one aspect that I want to stress compared to the blood plasma. So these guys are exposed to the blood plasma. And this is highly buffered. So buffering means that the as you rise in H pluses in that fluid, right? In the you now know that due to the Bohr effect, some of those H pluses could be bound to hemoglobin. Most of those are probably made by carbonic anhydrase out in the tissues, but if any of them come into the cell, they could be bound to hemoglobin. They could be bound to albumin or any of the other plasma proteins. So the blood is really quite buffered, but the cerebral spinal fluid doesn't have a lot of proteins dissolved in it like blood plasma does, and it is unbuffered. So as you go and let's see if I put it up here, it's too far away. As you go and look at this equation, let's say in the brain itself or in other body tissues, you are doing metabolism. And that metabolism raises CO2 levels in the cerebral spinal fluid. As it raises CO2 levels, don't worry about the carbonic anhydrase here. It's going to combine with water in the cerebral spinal fluid, and it's going to generate H pluses in the cerebral spinal fluid. Now, granted, slowly because we're not dealing with carbonic anhydrase, but it's going to happen slower than it would in a red blood cell. However, these central receptors are exquisitely sensitive to pH changes. So even if it's happening slowly, they are extremely sensitive. So let's emphasize that. So extremely sensitive to pH, to increasing concentration of H plus. Now where that H plus came from is from CO2 crossing the blood-brain barrier by simple diffusion. H plus cannot get through the blood-brain barrier. So any H plus that's out in the plasma doesn't directly talk to the central receptors. And that CO2 goes through combines with water to make HCO3, to make H plus. And of course, bicarbonate as well in the cerebral spinal fluid. Because these are so extremely sensitive to this increase, what you're going to learn in the negative feedback is that this is going to control 70% of your change in respiratory rate and depth. So as metabolism hopefully is going up in your brain right now, and you're making more CO2 because your brain cells are really active, then you may be increasing even slightly your rate and depth of respiration to try and get rid of this excess acid in your CSF. Are we okay with that? Any questions on that? So what's happening out in the periphery? So if you were to say, out in the periphery, start making CO2 as well, most of that CO2 is going to get taken up through the red blood cells and converted to bicarbonate, and the H plus is going to be bound up by the hemoglobin. But some of it, as you now know from these numbers, 7 to 10% of it is a dissolved gas. And that dissolved gas can do the same thing. So as PCO2 rises as a dissolved gas in your blood plasma, it's going to increase. That's going to be the primary and first way, aortic and carotid chemoreceptors are going to, can't talk and write at the same time, are going to detect this first. Whereas here, that rising CO2 causes H plus to go up. And this is going to constitute about 30% of the reason why you may increase your rate and depth. So it's the extreme sensitivity of these to H plus and therefore to rising PCO2 from PCO2 increase that makes these be more responsible. I don't think that is stressed in the book very clearly. This guy is not going to use H plus first because most of that H plus is going to be buffered either by the the plasma proteins or if it's going through carbonic anhydrase by the hemoglobin and the Bohr effect. And that's going to mean that it's the dissolved gas level that is going to be driving the increase because these guys are primarily detecting that. Are there questions on that? Okay, so let's see if, before we leave this, what else, I think you may have mentioned it, but what else is are the peripheral receptors? You said they are also sensitive to H plus, right? If I remember rightly, you said that, yeah? Now, why this is secondary sensitivity is because of all this buffering. Hopefully that's clear. So these are under normal circumstances in a person. If you start exercising intensely and you start generating lots of CO2, then you're also going to be increasing acidic levels in your plasma. Those are going to work together, those primary and secondary ways, to increase your rate and rate of breathing and the depth of breathing. And they're going to work with your central receptors. All three of these signals are going to work together to try and talk to the VRG to tell the VRG to increase the rate and depth of breathing. However, this guy also has a weigh station, which I think we're going to talk about on the next question, because it doesn't go directly to the VRG, whereas this guy is going to go directly to the VRG. Now, before we leave this to help you further, there's a third sensitivity, but it only happens under extreme circumstances, and that is low PO2 in the plasma. So for review purposes, what should in oxygenated blood, in arterial blood? Remember, we're looking at the carotid and aortic bodies. They're looking at blood coming right out of the heart, the oxygenated blood going to the rest of the body. So when you're in fully oxygenated blood, what's the normal PO2 levels from external respiration? Anybody can just say. It's in millimeters of mercury. Studying hasn't happened yet. Your list of multiple choice, 45, 100, 400, or what was the other, 40?
100.
100, right. 100, or sometimes we said 104. It should be that. Even if it drops to 80, which is, if you've been following COVID, you know that they're worrying about anything that drops under 90. So you don't want that to happen. This will only be active. This guy will only be active if it's less than 60. So you're in big trouble if your millimeters of mercury in your arterial blood is less than 60. So they're not very sensitive to oxygen, but under dire circumstances where your blood oxygen level is low, then they're going to increase their being stimulated by the high CO2 and the low pH. That's going to only be additive. They will be sensitive. What about the central receptors? Unfortunately, no, they're not sensitive. It's just your brain becomes more anaerobic and it makes more acidic waste under low oxygen, and therefore, they may get more stimulated by the H pluses. So this is a good start to central and peripheral receptor sensitivities. What are they looking at? What are they detecting? How are they working together? Who is more, in a sense, sensitive, which is the central receptors, therefore more responsible for driving the VRG. But the next question talks about the various roles of the VRG and DRG and the pontine respiratory nuclei. So let's go back to this anatomy again and share that one. And then who is the spokesperson doing 24?
It's me.
Who is me, Dina?
Yeah, sorry, Dina.
Okay.
I have a very long answer to it,
Okay.
and I'm not sure if I just rambled on into another question or what. But so what I have is that the respiratory rate and depth are set and regulated by the medulla oblongata. And then within the medulla, the ventral respiratory group, or the VRG, and the DRG work in conjunction to control and regulate respiration under the range of normal and abnormal conditions that affect respiration. In addition, the pontine respiratory center facilitates this process by smoothing out transitions between inspiration and expiration, as well as communicating impulses to the VRG that modify and fine-tune breathing while singing, speaking, sleeping, and exercising. The VRG extends into the ventral brainstem to set the rhythm and integration of respiration. It contains two separate groups of neurons that work in a mutual inhibition to create respiration. Inspiratory neurons fire to send impulses along the phrenic and intercostal neurons. These neurons excite the diaphragm and external intercostal muscles. The diaphragm descends and the thorax expands to increase lung volume while pressure in the lungs decreases as the air rushes in. When the expiratory neurons fire, they signal the cessation of inspiratory impulses to relax the diaphragm and external intercostals. Diaphragm ascends, thorax settles, and the lungs recoil, and expiration happens. The DRG integrates the input from peripheral stretch receptors and chemoreceptors to communicate this information with the VRG. The pontine respiratory centers are able to modify and influence the activity of the medulla oblongata and its BRG-DRG groups via the information received from higher brain centers and peripheral sensory data. To this end, the central and peripheral receptors trigger the negative feedback control of respiration rate and depth. Central receptors are part of the CNS and drive approximately 70% of the respiration rate. Due to the blood-brain barrier, the central receptors are not sensitive to changes in pH in the plasma, but they are very sensitive to CO2 levels, which are capable of crossing unbuffered into the cerebrospinal fluid. Changes in respiration rates have immediate effects on the amount of carbonic acid in the blood. When the rate of respiration slows and depth decreases, CO2 increases, carbonic acid increases, and pH drops. The opposite occurs with rapid, deep breathing, and these changes in respiration provide for quick action in the adjustment of pH and PCO2 in the blood. Conversely, the peripheral receptors or carotid bodies are highly sensitive to the chemical composition of the blood, responding first to PCO2 and then pH levels. They are less sensitive or less likely to be affected by PO2 levels, which need to be below 100 millimeters of mercury to affect them.
Okay. All right. So that was a lot of territory she covered. So let's recap. There are these two nuclei. Now, this ventral one, has motor neurons in it. These are actually, voluntary somatic motor neurons because they're going to skeletal muscles. Your diaphragm, remember we studied that in 103A, and the external intercostal muscles that are causing you to do your relaxed tidal volume breathing, those are all skeletal muscles. So you have voluntary control to drive these, because if you chose, you could stop breathing for a little while at least. So don't get confused, especially in the lab this happens, between what is unconsciously controlled and what is involuntary controlled. So when you are breathing and not thinking about it, which most of the time you're not, that's not because it's involuntary muscle and involuntary. It's because you can do this unconsciously. So like Dina said, within this group, there is a circuit of motor neurons that causes excitation to cause contraction of the diaphragm and the external intercostals when you inhale. And that occurs for around 2 seconds, if I remember right. You get excitation. And this is all involuntary, I should say unconscious. You're not worrying about having to think about every single time you go to move, just like if you're thinking of postural muscles, they're also unconsciously controlled. And you have to think about holding your back straight or your head up, or whatever posture. But if you want to change that, you can. It's voluntary. And then they stop firing so that you can relax these muscles and exhale for about 3 seconds. So that's going on in a rhythmic fashion without any inputs. You're not worrying about any inputs here. What's coming up from the central receptors all through the brain stem would be detecting the pH of the cerebrospinal fluid, which, as she and I mentioned, is exquisitely sensitive to PCO2 because that CO2 gets converted into H+. When you become a little more acidic, that is going to directly excite these motor neurons from the central receptors, and you're going to start increasing the action potentials going down to the diaphragm and the intercostals, and you may even start recruiting your PACs, your sternocleidomastoid, your scalenes, to cause a deeper breathing called hyperventilation. So that's the ventral respiratory group. The other two nuclei are like modifiers of this motor pathway. And as she mentioned, the dorsal respiratory group is receiving all peripheral inputs. So one of those peripheral inputs, or a major one, is coming from your carotid and aortic body. So as you're comparing and contrasting the central and the peripheral receptors, the peripheral receptors can't go directly to the VRG like the central ones do. They go to the DRG first, and then the DRG influences the VRG. So when your PCO2 levels are rising in your blood, they are going to cause CO2 to cross. The central receptors are going to quickly act on the VRG. The aortic and carotid body's peripheral receptors will also get excited, but they have to go to the DRG first, that then influences the VRG. Now, a lot of this is still being worked out and not fully understood. So this is the way we understand that now. Now, there are other peripheral receptors that also go to the DRG besides the carotid and aortic body. And you'll see those in the last slide in the series. So some of them are stretch receptors from your skeletal muscles, not just the diaphragm and the intercostals, but from your indirectly, multisynaptically, from stretch receptors in your muscles of your legs and your arms, so that as you're more active, that reinforces through the DRG that you are going to want to breathe deeper and faster, hyperventilate. Your pontine nuclei, as she mentioned, is chronically influencing these to smooth the transition between inspiration and expiration. And so sometimes damage in this pathway leads to a pattern, a characteristic pattern of breathing called apneustic breathing. Don't worry about that vocabulary word. But if this is disrupted in somebody who is just doing normal tidal breathing, its pattern will sound different. And that happens in brain damaged people. So it's got a term, that's associated with that kind of pattern of breathing. It also, when you are having descending inputs, you're emotional from your hypothalamus, you consciously want to stop breathing, you want to sing, you want to talk, you have to change your pattern. So these descending inputs coming from higher centers go to the pontine nucleus, and it influences the DRG and the VRG to enable you to change your pattern of breathing. So that's another long answer. You've just heard Dina and then me kind of walk through that covers 24 and touches quite a bit on 25 as well, the last question. So the last question puts this together as a actual negative feedback loop. Maybe we'll use this picture again, even though I can't write on it. So is there anybody, is that you still, Dina, talking about 25 or?
We actually just didn't feel like we came up with a good answer for 25 at all.
I can actually answer it.
Okay, go ahead, Noah.
So when arterial PCO2 increases, as we said before, it's gonna decrease the pH in the brain's extracellular fluid. It crosses the blood-brain barrier. And because of this, it's going to stimulate the chemical chemoreceptors that are responding to the increase or decrease in pH. And this will mediate 70% of the response. Then it's going to go into the VRG directly.
Right.
And then the peripheral, and then it's going to go and act upon the respiratory muscles to increase ventilation. And this is going to increase the rate and depth of breath. And this is going to return to arterial PCO2 and pH to normal. With the peripheral chemoreceptors and the cortotic and arthrotic bodies. Same thing, pH and CO2 is what's being detected here. And it's going to act upon the DRG and then to the VRG and then into the effectors. And then the other stimulatory or inhibitory stimuli, which I think is on your next slide.
Yeah.
I have to find it on mine. Yeah, so I mean, there's a bunch of things that can stimulate or inhibit. You have other receptors like emotion from the hypothalamus. You have higher brain centers for voluntary breathing, and both of these can either be excitatory or inhibitory. You have your central and peripheral chemoreceptors. You have receptors in your muscles and joints, which are excitatory. And you have your stretch receptors in your lungs, which are inhibitory, and irritant receptors that are inhibitory. And the DRG will integrate all of these.
Great. So there's a couple criticisms. I probably made them in the lecture with this figure and with the previous one. Like, he did a good job of separating VRG. They just lump it together here. Remember, the central receptors, like he said, are going directly to the VRG. The peripheral receptors have to go through the DRG to talk to the VRG. And it's only the VRG that are actually sending out axons to go to the respiratory muscles as effectors. The other criticism again here, and I think this arises partly, why they do the figure like this is partly because they realize we don't have full understanding of this. But if you see a receptor that's out in the periphery, so the joint muscle and joint receptors, the irritant receptors, the stretch receptors in the lung, the peripheral chemoreceptors, these all have to go through the DRG. It's only the central chemoreceptors that are going directly to the VRG. The other criticism I have of this figure is they really ought to put these in order, or maybe even just put O2 in parentheses, because the likelihood that you're going to have low O2 stimulate peripheral chemoreceptors is very low, since you have to really decrease the arterial blood's PO2 levels before that would kick in. H plus will, but your blood should be very buffered. Now, acidosis is a not uncommon event, but it's not going to happen with normal exercise or normal activity. We will be talking about having low pH and high H plus levels in your blood, but those are usually in pathologies. They're in an abnormal situation. So really, your peripheral receptors under normal conditions are running on PCO2. And the central receptors are running on H plus, indirectly PCO2. So putting both of these, it's almost like there ought to be an arrow between the CO2, which causes a rise in the H plus. So again, the diagram is got its limitations.
Professor, I actually had a question.
Yeah.
I was reading the book to further understand the question, but it seems to separate this negative feedback loop from exercising, saying that they're sort of two different things, as in exercise-induced ventilation, which would be hyperapnea or apnea, is different from hyperventilation caused from hypercapnia, I believe. So that's why I was confused about my group was confused about the question at first, because it's talking about exercising. So I don't know if you could clarify.
Well, I'd have to look at exactly the wording that they have in the book.
Well, they say because exercise enhanced ventilation, like during exercise, pH, CO2 levels and O2 levels and pH sort of say neutral. They're like constant, whereas with hyperventilation, you need low PCO2 to cause excess hyperventilation, I guess, is what the book says.
Where are you in the book? Maybe if I look at it.
Page 857.
7. And under normal conditions, declining PO2 has only slight effect on ventilation. Is that where you are? Which paragraph?
It is like labeled exercise. I don't know. It's literally labeled exercise.
Oh, I see. It's over here on the other side. I'm looking at the wrong page. Okay. Working muscle tremendous amounts of O2 and produce large amounts of CO2, hyper.
And then for comparison, it talks about hypercapnia,
Okay.
like 2 pages
So
up.
they're making a distinction from what I'm gathering, reading this quickly, between somebody hyperventilating. So you're sitting in a chair, and we will come back to this when we do acid-base balance. You're sitting in a chair and you have a panic attack, because I just told you the exam is actually today, and you start hyperventilating. So you're starting from a normal level of CO2 in your blood and a normal pH in your blood, and you're expelling excess. And that's causing your blood pH to actually rise, not fall. So it will be somewhat different. We'll talk about this more with acid-base balance. Versus as you exercise, first of all, you're going to increase your sympathetic, so it's going to go through these pathways, and you're going to be basically trying to keep that PCO2 level that might rise otherwise. You're trying to keep that within its normal boundaries. So is this helping? No?
Sorta. I can always ask it in office hours just to clarify.
Yeah, I mean, I wouldn't get too stressed about this section yet, because we will come back to these ideas when we talk about acid-base balance. And I
Okay.
think that's the problem here. You know, they're bringing in lactic acid, things that we're not going to worry about yet, because we're going to return to this stuff. All right, before we leave this, I would like you to know this reflex, understand this reflex. And it's not going to happen very much, but it does have a name. And so it could show up on the test Thursday as a name. And that's that the lung itself, and I'm not sure where in the lung it would have it, maybe in the smooth muscle of the bronchi. I don't know, though, where you're trying to protect your lung, because after all, it's thin tissue. You know, the alveoli are very thin and delicate tissues. Now, I don't know when this would happen, that you could overstretch your lungs, but I guess maybe if you're using a CPAP device or something and they're really forcing a lot of air into your lungs. So there is a reflex that depresses the respiratory muscles so they don't contract as hard and try to protect the lung itself. So that's called the Herring-Brewer reflex. Okay, any questions on this? And we're almost out of time, I can see that. Any questions? Oh, you've got all the answers as much as you can on the on these lecture questions. All right, so we have about 10 minutes left, which I know isn't a lot for review. Let me stop sharing this. And then see what you guys would like to do. So I mentioned, for those of you who are late, when you come in, you will see the format of the test, and that you should show up. You should show up a few minutes early just to make sure, but it will open and become available to you. I do encourage you, if you can, to be on Confer Zoom at the same time you're in Canvas to take the test, because I think it's the easiest way for you to ask questions. I'll be on Confer Zoom working while you're doing that, and I'll be glancing up at chat. So just put the questions onto chat. I'll try to have my other screen open to the email, but I only have so many eyes in my head. So paying attention to chat, paying attention to my email, and then, quietly doing my own work will be hopefully get you an answer quickly, but I want to make sure you do get those answers. Okay, so what does anyone have suggestions on what you'd like to do, or specific questions for review? And then we could do more review in the lab if you'd like to also. Does anyone have any questions? No? You want to just take a longer break and then come back and we could do some review in the lab. The lab time otherwise is scheduled to be time for you to finish up the histology slides if you haven't finished them, and for us to go over the spirometry handout. But other than that, I don't have anything scheduled for the lab. How about people, if you want to, let me get it in the reactions. Give me a thumbs up if you just want to take a break for 20 minutes, get your lecture. Okay, it looks like there's a massive number of people. And in that time, you can come up with what you want to do for the review, if you'd like a review. All right, it looks like I have some non-voters, but we have a lot of thumbs up voting that you just would like to take a longer break, think about things, and come back with some questions. Okay, so I will see you at 11:10. We'll end this a little early, and see you in lab. And if you can't make the 11:10, I'll see you at 1 o'clock. Okay, all right, we'll see you later.